reminds me to turn on the recording. Make sure I don't forget to do that. All the previous talks have been recorded. You can find all of those at the Innovate Bio website, as well as the talk schedule. I already mentioned the last 2 talks of the series, and I also want to mention too, we are going to have 2 special webinars in May. On May 6th, we're going to have an hour-and-a-half webinar session with breakout rooms that Dr. Celeste Carter is going to join, and we're going to be talking about best practices in running college biotech programs. So that should be a lot of fun. And on May 13th, Dr. Lisa Seidman and Dr. Jeanette Mowry are going to join us and talk about the new Bible, you might say, for biotech lab courses. And you may have seen the textbook that Lisa's written, but there is a new edition, so it should be a lot of fun. The format for these talks, we have our, the 2 talks are going to be sequential, and we'll have discussions and an evaluation at the end. We really want you to fill that out because that helps us with informing what we do and providing feedback and accountability to NSF. Our goals here are to showcase the advanced technological education projects in this series and help people know what other people are doing and what's involved. We also have a Slack group, which we're kind of learning how to work with still, and we'd really love it if you joined and start talking on Slack, which it's a lot of fun. I have an invitation here, and it is good for 9 days, but I can always make new invitations, and I will put this in chat in just a moment. Okay, I think without further ado, I'm going to stop screen sharing, and Joseph, it's all yours.
All right, thank you very much. And can I just get a thumbs up that you guys can see my screen before we start talking? All right, fantastic. My name is Joseph Oleniczak. I am the principal investigator for this NSF ATE project, and I have 2 affiliations. I'm with Austin Community College in the biotechnology department there, and I'm also on a high school campus, and the high school campus I'm on is the Liberal Arts and Science Academy. And a little bit about myself and my program. All of my courses are dual credit. So these are courses where students are enrolled in high school as 11th and 12th graders, and simultaneously they're getting credit through Austin Community College in the biotechnology program. My co-PI will introduce himself now.
Yeah, I'm Kissaou Tchedre. I'm a full-time faculty at Austin Community College in the biology department and biotech. So I teach both biotech courses like 1414, 1415, and many other courses. Yes.
Excellent. Well, thanks again to Innovate Bio for hosting us today, and we're excited to share about our project. Our project is a dual credit high school community college DNA sequencing and genomics project. So I'm going to talk a little bit more about what we're doing this year and sort of where we plan to move moving forward with this particular project. So the overarching goal of the ACC Biotech program, which I'm sure is similar to a lot of other biotech programs out there, is to just simply increase the number of dual credit high school as well as traditional 2-year students who enter into the program, complete certificates and degree, and eventually get into the workforce. At ACC, we offer a level 1 certificate, which is the fast track certificate, an advanced technical certificate, which is a post-baccalaureate certificate, and lastly, an associate's degree. Today, from the high school perspective, I'm going to talk mostly about the level 1 certificate because we've actually embedded that into my high school dual credit program. And part of this project is really aimed at trying to get more students to complete the certificate, and after they've completed that certificate, to move into gainful employment in the biotechnology industry here in Austin, Texas. So the specific goals for our project there are 4 goals. Number 1 was to establish a DNA sequencing and genomics facility with industry-level DNA sequencing technology on our dual credit high school campus. 2 was to develop course curricula that with a focus on authentic student-led research using DNA sequencing that was not only taught in some of our introductory courses, but in later courses in our sequence, mentored by local industry and academic partners. 3, part of the mission that ties into sort of the mission of our ACC Biotech program was to increase the recruitment, retention, and certificate and degree completion rates for all of our students with an emphasis on minority students in particular in our training program. And lastly, what we're doing today. which is to disseminate the work that we're doing with the Innovate Bio community. We are in year one of this 3-year project. We kicked off in May of 21, and we're about to come up on the end of our first year. I mentioned it earlier, but my dual credit program is really geared at getting students to complete the level 1 certificate through Austin Community College and then move into employment in local biotechnology industry. So just to kind of give you a little bit of context, that certificate and the courses associated with it are shown on the slide here. Most students that I interact with are in these 2 courses here, Biology 1414 and 1415, which they take ideally in the fall of their 11th grade year and in the spring of their 11th grade year. And then if they continue in the program, over the summer, they take a quality assurances in the biosciences course, which is an online course. And in year 2 of the program, they take a capstone course called BITC 2486, which is an internship course where students normally go off campus to work in research internships and local biotechnology, either industry labs or at the University of Texas, which is also here in Austin, in a biotech lab there. Really, the goal of our project was to see if we can recruit more students into this 1414 and 1415 course sequence. And once they've been recruited into this course sequence, how can we get them to continue in the program towards 1340 and eventually this capstone's course 2486 to complete that level 1 certificate. Just a little more background information about this. Sort of the big hurdles we have with students transitioning from these introductory courses and into the summer course and into the internship is simply providing internship opportunities, especially for high school students. Their schedules are very busy, so finding time outside of the school day can be a challenge, and also finding internship placements that will accommodate students under the age of 18 can also be a challenge. So by establishing the DNA sequencing and genomics facility here on our high school campus, we hope to kind of overcome these challenges and provide more opportunities for students to segue from these courses to the rest of the course sequence here. A couple of key activities when it comes to our specific project. In year 1, we procured, installed, and validated an applied biosystems Seq Studio genetic analyzer here on the LASA campus. So we've established our sequencing and genomics facility in terms of instrumentation. We decided to go with this particular sequencer because most of the DNA sequencing we're doing on our campus is related to DNA barcoding. So this is an automated Sanger sequencer, which is ideal for short reads that you're dealing with in DNA barcoding. 2, this year we worked with some partners up at Cold Spring Harbor DNA Learning Center. They have a well-established curriculum for DNA barcoding, and we've been using versions of that for introductory DNA barcoding labs in that Biology 1414 course that I mentioned earlier. We've sort of adapted some of that to make it more specific to our campus. And in addition to that, we've added in the component of doing in-house sequencing. So for a lot of these DNA barcode labs that we've done in the past, students would go through the DNA barcoding process, which is simply bringing in a sample, extracting DNA, amplifying it using PCR, cleaning up that PCR product, and then sending it out to another core facility to be sequenced. Our NSF ATE project really focuses on students being able to sequence it in-house on campus. And not only do they get to learn more about the sequencing process as a result of that, they also gain some of those instrumentation competencies that they can translate into workforce by learning to actually use the sequencer itself. This year, we also piloted those protocols that I just mentioned with 94 of my dual credit students here at the LASA campus. And Kissaou did that with 8 of his community college students who were actually here last night on our high school campus using our DNA sequencing facility here. Lastly, we're working towards building an academic and industry mentor network for our students who want to do DNA sequencing and genomic projects next academic year. So I'll back up 2 slides, or back up 1 slide, excuse me, just to explain that. So I kind of mentioned earlier, we recruit students here into 1414 and 1415, but we really want to emphasize if you're taking these courses, you should continue in the program. A lot of my students at the dual credit high school level, again, have a hard time finding internships off campus or don't have time in their schedule. So by learning about DNA sequencing through barcoding in their 11th grade year, they could do DNA sequencing and barcoding projects here on campus in their 12th grade year that are mentored by outside industry as well as academia who come to our campus to mentor these students. So this year, like I mentioned, we have piloted all of our curriculum as well as our sequencing facility with our students. And I wanted to walk you through some of the pre and post survey data that was collected from our dual credit as well as our 2-year students. So we're going to start over here on this left-hand side of the slide. Before students went through the DNA sequencing module that we developed, we had them rank their understanding of how well they understand how DNA sequencing works, using DNA sequencing instrumentation, executing laboratory protocols for DNA sequencing, and lastly, for interpreting DNA sequencing data. And this was self-ranked data from no knowledge to beginner to proficient, advanced, or expert. And as you can see in the pre-survey data, most students fell into the category of knowing very little about DNA sequencing, how to use the instrumentation, or how to interpret those results. The post-survey data tells a very different story where almost all students are shifting over into this advanced or expert category, which was evidence to Kissaou and I that the curriculum we're using is effective at getting that background information to students about DNA sequencing, and having that hands-on component of them actually using the DNA sequencer is making them more comfortable with using these protocols and understanding that DNA sequencing instrumentation. Really, and this ties to our project goals, and I wanted to kind of highlight this survey data. Over here on the right-hand side of the screen, if we look at C and D, we asked them a question beforehand, doing the DNA sequencing module, how likely they were to continue in the program to work towards that level 1 certificate. And as you can see, students ranked themselves from 1 to 5, 5 being they were very interested in completing the certificate, 1 being very not interested in completing the certificate. And the data was pretty well spread throughout those categories. The post-survey data tells a very different story. We saw a huge shift to most students over here in the 4 and 5 category, really wanting to continue in the program as a result of having this experience. So what does that mean in actual student numbers? I just wanted to highlight a couple of things from my program, and then I'm going to hand it off to Kissaou to talk about things from the traditional 2-year program. For this year, whoops, excuse me, I went 1 slide too far here. Let me back up. For dual credit students, next year we have 55 students who are going to continue in our program and work towards that level 1 certificate. And this is a huge increase from this academic year, which only had 20 students this spring who are going to be completing that certificate. In terms of recruiting students into the program, this year we have 94 students who took that introductory 1414 and 1415 course sequence. Next year we have about 128 students who have signed up for it. So another marked increase in the number of students moving into the program. Just sort of anecdotal evidence too. I'm in the biotechnology room, day in, day out here on the high school campus, and really bringing in the sequencing facility has generated quite a buzz here on campus with students who are not even in our biotech program stopping by just to sort of, peek in and see what's going on in the biotech room. In addition to that, I mentioned the research mentor network that we're going to work with next year. We've recruited 6 industry mentors from Thermo Fisher, Asuragen, and PerkinElmer, 3 mentors from the University of Texas at Austin, and 3 mentors from Austin Community College. And the intention there is for students who can't go off campus to complete that last capstone course in the internship, again, off campus, either in industry or at a university lab. They can do it on campus, doing either a DNA barcoding project, a DNA variant analysis project, or some other project related to DNA sequencing that we can accommodate using the instrumentation and reagents that we have in our lab. At this point, I'd like to pass it over to Kissaou, and he can speak to the 2-year portion.
Yes, okay. So for the first year for the 2-year program at ACC, so we had 8 students who were enrolled in biology 1414 and for their advanced certificate. Okay, so compared to 5 students last year, and then all of them are involved in these projects. And like Joe just said at the beginning, yesterday night they were at the sequencing genomics center to run their samples from the DNA barcoding lab. So you could see even from them the way they ask, they participate, they ask questions. You can see they were really interested. And when we mentioned to them that they were the first promotion to sequence their own barcoding samples, they were very impressed about it. So we can see it generates some interest in students, and then we expect to increase the number as things are going. And for the coming semester also, the plan is to increase at least the number of students up to 40 to 50 because we plan to introduce also the same DNA barcoding in the biology program, which is a biology 1406, which has more students. So we plan to add the same program, but there will be barcoding moth samples, and together they will use those samples and then send them to the Genomics Center for sequencing. So with this is going to increase our number, and then we should be able to really incorporate this program so that the student can take advantage and then develop their sequencing skills. Well, in the future, also, we may introduce the DNA sequencing in BITC 2441, which is a molecular techniques. And in general, the Genomics Center is going to become very important for ACC and then LASA students. And we expect to really generate and then increase student skills in sequencing. And who knows, maybe down the road, we can introduce also some whole genome sequencing for them. So this is basically what we have planned for this coming year for 2-year student at ACC.
Thanks, Kissaou.
Thanks.
Lastly, we wanted to end on a couple of photos to sort of showing the sequencing center in action. So these are some of my dual credit high school students over here setting up their 96 well plate before they load it into the sequencer. This is them loading it into the sequencer here and just sort of interfacing with the sequencer itself. Like Kissaou mentioned, these other photos are literally from last night. He had his 2-year students come to our high school campus and prep as well as load their samples into the sequencer here on campus. So these are photos from that activity last night.
Last night, yes.
And lastly, that sort of concludes our presentation for the day. I look forward to hearing the next presentation, and I really look forward to the Q&A. I hope there are a lot of questions for us, but I also have a lot of questions for the group. We're sort of thinking down the line. We're in year 1 of a 3-year program. We've been very successful this year in establishing the center, recruiting students, and moving towards getting more students to work towards that level 1, and as well as other certificates and degrees at ACC. But we really want to talk about how we can scale this program, how we meet some of those needs, and after the grant ends in 3 years, how we can make this a long-term thing. As you know, buying a sequencer and all the reagents to run it is very expensive. So we're curious to hear from the group if they have ideas. So for moving forward and keeping this in perpetuity here as part of the LASA ACC program.